Well, thank you very much, Juan, for the nice introduction, and I'm not sure I deserve it. And thank you for the organizers for having me here. It's been really a very interesting meeting, very high quality, and despite the weather, I haven't seen this much rain in my life. So uh, I think the place is really great. I'm also very happy to talk at this meeting that is entitled after Dr. Ina Win Arias. My mentor, Professor Gentilini, was a very close friend of Win, and although I'm not completely sure which are the reasons why it's entitled after him, I'm really very happy that he's mentioned in this symposium. So actually, when I looked at the title that is Bridging the Gap Between Basics and the Clinic in FLD, this is a very wide topic. And I think in terms of what is going on in therapies for an FLD, Dr. Torre in one of the previous lectures actually has very well made the point on what are the ongoing data. So I have replaced some of the slides and I will actually go very fast, trying not to repeat what has been already told today. These are my disclosures. And we have already seen the slides, I mean, part of it or some of it. And this is after a recent review that has been published in the New England Journal, showing that you can follow the pathogenesis of NASH from the initial metabolic damage that leads to the generation of a fatty hepatocyte to the essence of NASH, that is the ballooning, that is the hepatocyte damage, that leads to lipotoxicity and hepatocyte apoptosis, lipoptosis, or even necroptosis. And these are signals that eventually generate activation of cells which are involved in the tissue repair, including the myofibroblastic stellate cells, duct epithelial cells, sinusoidal epithelial, endothelial cells, and immune cells. So we can see the pathogenesis of NASH as a sort of like working wheel that leads from simple steatosis, that is the metabolic damage, to portal inflammation, NASH, and fibrosis. And we can dissect the possible areas where we can intervene with drugs with the metabolic injury, cell injury and inflammation, and fibrogenesis. And I will share with you some of the data that we are currently generating in my lab according to these three different topics. And I will also do reference to what has been published and what is actually going on. You can see that in the same issue of the journal, there is this table where the formerly used and drugs under investigation are differentiated according to their targets. You can see that according to the metabolic stress, inflammation, and fibrosis, there is a miscellanea of action. And for example, you can see galectin-3 has been nicely shown by Dr. Torre that works mostly on fibrosis and inflammation, whereas other drugs such as the obeticholic acid acts on all three the hits. So if we look at the pathogenesis of fatty liver, we know that the fat may come from dietary components through de novo lipogenesis and through lipolysis of the adipose tissue. This is actually the major component. And this leads to oxidation by different organelles within the hepatocytes, generation of ROS, or accumulation of triglycerides that may be stored as lipid droplets or secreted as VLDL that eventually lead to atherogenic dyslipidemia. So for example, FXR works on several of these pathways, such as, for example, beta oxidation or other lipogenic pathways, and it's in the strict association with the gut-liver axis, because FXR also works at the intestinal level, where it contributes to the release of FGF19 in humans and 15 in the mice, that acts again in the hepatocyte through beta-klotho and the FGF receptor 4, leading to amelioration of steatosis and metabolic factors. And this is just a slide summarizing how FGF19 works. You can see here the FGF4 receptor, and at the same family belongs the FGF21, that is actually a hepatokine. It's a cytokine that is generated within the liver as much as the sirtuin that was referred to earlier in the talk of Dr. Armendaritz. So these have been shown. These are very promising therapeutic approaches, such as the modulation of the GLP-1 system that can occur mostly through GLP-1 receptor agonists, like in the recent lean study where liraglutide was used in a small number of patients with NASH. These factors also work very well inducing body weight reduction, and they have been approved or are going to be approved for the treatment of obesity, and especially the new generation like semaglutide is very promising also for the treatment of NASH. So if you look at some of these recent reviews like this one that has been published in Journal of Hepatology, again, you can see that the different molecules that are under investigation are differentiated into their metabolic components, anti-inflammatory and antifibrotic. And I'm not going to repeat what has been said before in this symposium, just going through the different molecules. So cell injury and inflammation is another important component. Inflammation in NASH is really multifaceted because, there is inflammation in the adipose tissue that leads to change in the adipokine pattern. There is inflammation in the gut, leading to increased availability to the liver of pathogen-associated molecular patterns. And of course, there is inflammation within the liver through cellular stress and danger signal that may be generated also by dying cells itself, what has been named as a danger, so damage-associated molecular patterns that work through the same pattern recognition receptors. And finally, there is the modulation of inflammation. There are M2 macrophages and some other macrophages that actually are involved in the resolution of inflammation. So it's very important how to modulate these cells. And in fibrosis, again, there are M1 macrophages that are more immune activating and M2 macrophages that are more profibrotic, and then you have pro-resolution macrophages that have yet another phenotype. So let me show you some of the data that we are currently doing with this macrophage fibrosis axis, and this is a slide I already showed. Is like the TEM family of tyrosine kinase receptors that bind as a ligand GAS6, and these are the two mostly studied, MERTK and AXL-TK. They are expressed in hepatic stellate cells and in macrophages. So in collaboration with Salvopetta in Palermo, we published a couple of years ago that MERTK, this kinase that is expressed at higher level in the liver tissue of patients with NASH, is mostly expressed in macrophages. You can see here the immunohistochemistry, where the blue signal, the green signal in macrophages co-localizes with MERTK with the red signal giving a yellow signal. So in our lab, we struggled a little bit to differentiate the macrophages to express high level of MERTK, but finally we did that in cell lines like THP1 monocytes, and we are now repeating it in primary monocytes, that with a differentiation protocol using PMA, they express at very high levels MERTK, and this MERTK may be activatable by exposure to GAS6, that is the ligand. You can see here the phosphorylation-specific antibody showing very nicely phosphorylation of this molecule. So what we did, we took this monophage with these macrophages, we incubated with GAS6 in the presence or absence of a specific antagonist of MERTK, washed, and then incubate the stellate cells with the conditioned medium, trying to show whether exposure to these cytokines, so this GAS6 and activation of MERTK could be relevant for the biologic activity of stellate cells. And here you can see that these are the controlled, the migration of the stellate cells. If the stellate cells are just exposed to the conditioned medium of these macrophages, they increase their ability to migrate, that is a profibrogenic activity, but they do more so if the macrophages have been previously exposed to GAS6. And both these biologic activity are inhibitable when the macrophages, but not the stellate cells, are pretreated with the MERTK antagonist. And similar data we obtained for cell proliferation. You can see the layout of the cartoon is pretty much the same, and you can see here again that there is a very nice inhibition if you block MERTK. Interestingly, also the expression of genes that are involved in fibrogenesis, like TIM1, collagen 1 alpha, TGF-beta, and pro-inflammatory factors such as interleukin 8 are also modulated in this fashion. So we are actually elucidating an axis involving MERTK where lipotoxic damage induced in hepatocytes leads to activation of MERTK via Gas6 that activates stellate cells by itself. This has already been published, and I didn't show you this data. They are in Journal Hepatology. But also to the macrophages that through paracrine factors activate stellate cells. And we're actually now trying to identify which are these factors that are secreted by the macrophages. Another area in inflammation that we are currently pursuing is the activation of this molecule, this IDO, that is indoleamine 2,3 dioxygenase. This is a very interesting molecule because it has been involved in many conditions, including cancer, where modulates the immune system. So what we did, basically, this is the outline of the pathway. It's a pathway where tryptophan, the amino acid, is converted to kynurenine, that is a biologically active molecule that may be modified into some other metabolites like kynurenic acid, hydroxykynurenine, and quinolinic acid, giving a number of metabolites that are biologically active. And this pathway may be inhibited by like an antagonist that is a modified tryptophan is called 1-methyltryptophan. And we have used this molecule to see what happens in a model of NASH to the IDO pathway. And we found that if you have mice that have been treated with a methionine and choline deficient diet, these are the blue column, they increase the number of activated leukocytes. This is obviously known, but these leukocytes are present at enhanced levels when the mice are treated with 1-methyltryptophan, showing that if we inhibit the IDO1, the IDO system, you exacerbate inflammation. And you can also see increased expression of NOS2, increased expression of TNF1 alpha, TNF alpha, and increased expression of myeloperoxidase, that is a molecule that is expressed mostly in neutrophils. So inflammation is exacerbated, and this is associated with a reduction in hepatic kynurenines, that are the metabolites that are produced by the IDO pathway. As you can see here, compared to mice receiving vehicles, the 1-methyltryptophan antagonist leads to a significant reduction in hepatic kynurenines. We went ahead and looked at the crosstalk between this interesting molecule and another system that is very interesting in my opinion, that is the inflammasome system. We have already heard something about the inflammasome. And we we started from a previous study that we have recently published, so showing that a nutraceutical, that is berberine, it's a herbal compound, reduces damages in steatohepatitis, actually working through the inflammasome pathway. And we also noticed that berberine, going back to these mice, induces IDO1. So we had the hypothesis that part of the anti-inflammatory effect of berberine could actually be mediated by increased expression of IDO1. And this occurs both in the liver in vivo, but also in macrophages. Here, these are macrophages that have been challenged with LPS, and they already increase IDO1 expression, but mostly when LPS is coupled to berberine. So we went back to the mice that I already showed you, the ones that were receiving 1-methyltryptophan. And what we observed is that interleukin-1 beta, that is the final product of the inflammasome pathway, is expressed at higher levels in mice on the MCD diet with 1-methyltryptophan. And 1-methyltryptophan inhibits IDO, I remember this to you. And also components of the NLRP3 inflammasome, like ASK1, for example, caspase-1, and NLRP3 are modulated. Going in vitro, we looked at the macrophages again, and if you compare this column, so this black column with the light gray column, these are macrophages that have been challenged with submaximal doses of lipopolysaccharide. And as you can see here, if you treat the macrophages with the inhibitor of 1-methyltryptophan, you obtain a significantly higher expression of IL-1, increase the secretion of IL-1 protein, and increase expression of NLRP3, that is the molecule that characterizes this inflammasome. So in this study, we are establishing a novel crosstalk between the IDO pathway and the inflammasome pathway. And the inflammasome is interesting not just to us, but to many other investigators. This is from a recent review showing that there are several ways to interfere with inflammasome activation. For example, dopamine or even interferons may interfere at the level of assembly of the inflammasome component. And I think this is a very interesting, potentially new pathway. And this just summarizes the anti-inflammatory pathways. I already, someone already mentioned, for example, interfering with TLR4 or interfering with trafficking of the immune cells. Of course, fibrogenesis is appealing because we know that fibrosis is probably the most important component of the wound healing response, and is a major determinant of the prognosis of these patients. We already heard about the relevance of chemokines. I will be very fast with that. CCL2 and CCL5 are ligands of the chemokine receptor CCR5 and CCR2 that are targeted by cenicrevirok. Another approach, and cenicrevirok, as we heard, is already in phase 3 investigation, and similarly, ESK1 is a kinase that targets both Jun kinase and P38 and leads to activation of macrophages, but especially to induction of the profibrogenic actions of stellate cells. And also, in this case, selonsertib, made by Gilead, is in phase 3 investigation. So these are already examples of how the molecules that have been initially characterized in vitro and in preclinical studies are being currently and actively translated into possible clinical practice. Of course, we will see the results of the trials. An interesting thing is sarcopenia. Sarcopenia, we have heard a lot about sarcopenia, and even in this meeting, sarcopenia is linked to physical inactivity, but is also linked to the metabolic derangement of these patients. And we are currently investigating, I've already submitted the paper here, whether a myostatin, that is a molecule that contributes to sarcopenia, has a role in fibrogenesis. Myostatin activates the activin receptor 2B, and like other molecules of the TGF-beta family, works through activation of SMED2 and SMED3. And these are some of the data showing that the human hepatic myofibroblasts derived from stellate cells, their biologic features may be modulated by exposure to myostatin. And this occurs, as you can see here, in a Jun kinase dependent activation. These are cells where Jun kinase has been silenced with a small interfering RNA, and you can see that the biologic effects in this context, migration of the cells, may be inhibited when Jun kinase is silenced, and similar data we observe with Jun kinase inhibitors. So to make a long story short, I think we are sort of elucidating a muscle to fibrogenesis axis that could involve myostatin, and this leads to induction of TGF-beta, procollagen expression, and chemotaxis of the cells. So I think this is an interesting point. And again, antifibrotic factor molecules include also galectin, not simtuzumab, it was already tested, but unfortunately the trial was negative, and we can interfere also with this pathway. So in the final 10 minutes remaining, I will start from some observations that we have made in hepatocellular carcinoma, going back to what is going on in the NAFLD, the NAFLD area. So we know that obesity and NAFLD are associated with an excess prevalence of hepatocellular carcinoma that occurs not just in patients with cirrhosis, but also in patients with non-fibrotic steatohepatitis, and even in patients with steatosis and without fibrosis. And we all know, as clinicians, that is a major problem when we are going to put these patients under surveillance, because of course there are no surveillance recommendations made by any of the our favorite scientific societies. So we were interested in the microenvironment that leads to the development of HCC. Coming from the fibrosis area and the stellate cell biology area, we started looking at a number of soluble factors and how the soluble factors that interfere with fibrosis may also be important for generating an environment that is fertile for the origin of cancer in these patients. And we focused on a molecule that is, in our opinion, very interesting, although it has not been very actively studied. It's a member of the MAP kinase family. It's called ERK5. Like other members of the MAP kinase family, it is activated through a kinase cascade that involves a MAP3K, a MAP2K, and the MAP kinase itself. And it has a number of downstream effectors that include MEF2C, P90 risk, and other molecules. It's relevant for survival, proliferation, and apoptosis. If you make a knockout mouse for ERK5, this mouse is not viable. The mouse dies very early, I think at embryonic day 10 or 11, if I remember correctly, because of very severe alterations in the vascular and circulatory system. So we observed that if you take a cell line, these are HUH7, but we have done with other cell lines, and expose it to factors that are very high in the environment of the fibrotic liver, like epidermal growth factor, TGF-beta, or leptin, you observe a marked activation of ERK5. We have looked at that in the context of cancer. These are a considerable amount of data that we have recently published in GUT, showing that if you silence ERK5 in these cells, the cell cycle is altered, and especially the cells become more abundant in the G0, G1 phase and less abundant in G2 and the S phases. And more important, if you look in vivo and you make a xenograft of these cells in a nude mouse model of ectopic hepatocellular carcinoma, you can see that the number of animals that develop tumors is dramatically reduced if you inject cells that have been silenced for ERK5. And similar things we observe if you use a pharmacologic inhibitor that is administered to mice once the cancer has already established, and you can see how nicely the two curves diverge. These are the mice that are receiving the inhibitor that calls XMD 892, and these are the mice treated with vehicle. So what are we doing now? These are data that are published. Because we cannot really look at the genetic ablation of the gene, because as I told you, the mice are not viable, we generated mice with conditional mice. So we generated mice with hepatocyte-specific deletion of ERK5 under the albumin promoter, and we also obtained mice with myeloid cell-specific deletion under the lysozyme, the LysM-CRE promoter. And we're not going to talk about these mice, and we'll just show you some preliminary results regarding the possible role of ERK5 in hepatocytes, in liver regeneration, and the transduction of insulin signals and insulin sensitivity. So if you do liver regeneration, whereby the classical model of 2/3 partial hepatectomy, you see progressive recovery of the liver weight, and this slide just shows you the liver to body weight ratio. There is maybe a little bit of delay. We are now working in the early time points. There is a little bit of delay in the recovery of the weight by mice lacking ERK5 in hepatocyte. These are the orange columns, but as you can see, there are no major differences. What is impressive is that the regeneration of mice without ERK5 occurs at the expenses of very high, a marked elevation in aminotransferases, both AST and ALT. They are modestly increased in the control mice, but reach like 5,000 units per liter for AST and 2,000 for ALT. And this is associated with appearance of numerous and abundant foci of necrosis in the liver of this patient, of these mice undergoing hepatectomy with the genotype that gets rid of ERK5 in the hepatocyte. So the promotion that we see in cancer is maybe counterparted by the action of protection of these cells during partial hepatectomy. And of course, now we are looking at the molecular mechanisms that underlie this observation. Another model that we're looking at is the model of steatosis. It's just the model that Dr. Armendaris showed you, is the high-fat model for 16 weeks. And these mice develop, like increase their body weight and develop increase in blood sugar level. As you can see, again, here, the mice in orange are those without ERK5, and the mouse in blue are the control mice. You can see there are no changes in body weight. I can tell you there are no changes in food consumption. There are minor changes in fasting blood glucose that is actually a little lower in mice with the genetic deletion. But the important thing is that the amount of fat in the liver is increased in these mice. But what was actually very interesting is that if you subject these mice to a glucose tolerance test, you can see that the tolerance to glucose is much lower in mice which are lacking ERK5. And again, the area under the curve is statistically higher if you compare these mice with the control mice. And this would mean that ERK5 is implicated in insulin sensitivity, because if you delete it, and specifically from hepatocyte, you will observe higher levels of glucose. And this is also confirmed by the insulin tolerance test, where by a single injection of insulin, of course, glucose drops, but drops much less in the mice that have the genetic deletion as in those that do not have the deletion. And again, here the area under the curve is significantly different. So we're very excited about this project. We have a number of ideas of why this may be occurring. There are a number of molecular candidates that at this stage I cannot disclose because it's very, very preliminary, but that hopefully we'll be able to elucidate in the coming weeks and months. So concluding, I think there are several aspects that might be translated. This is a very active field. I was just in a pause from the rain. I was in one of the markets here in Cancun, and we have already several molecules that we can possibly translate. We have molecules for the complications of NAFLD, like rignonina and diabetina, and that are well-known associations with the with NAFLD. But I was actually impressed by the tel dell'indio, because the tel dell'indio, you cannot read it, but it's an auxiliar anhigato grasso. So I actually already bought a couple of kilograms and bring it back home just to make sure we can translate it in the future. Thank you.